Good afternoon. We welcome you to the Conference on Plant Epigenetics. This activity is offered to you as part of the Meet the Expert series of the National Science Foundation project at the Interamericana University of Puerto Rico, Barranquitas campus. We acknowledge the presence of Dr. Juana Negrón Berríos, Chancellor of Barranquitas campus, Dr. Alok Arun, Director of the National Science Foundation project, Dr. Nidia Rodriguez, Co-Director of the NSF project at the Guayama campus, Dr. Mary Gelling, guest speaker from the Whitehead Institute and the Massachusetts Institute of Technology, professors, administrative personnel, and students of the Barranquitas campus, Guayama campus, and other campuses of the Interamericana University of Puerto Rico, students of the Bilingual Academy of Barranquitas, other guests from and outside Puerto Rico that accompany us in person and virtually. Thank you all for being here with us this afternoon. As it is a custom in our institution, we will begin this activity with an invocation led by Reverend Arnaldo Cintron Miranda, Director of the University Chaplaincy. I kindly ask you to stand up.
Muy buenas tardes a todos y a todas y a los que están también virtuales. I'm the speaker de esta ocasión. Welcome to Puerto Rico, the campus of Barranquitas. Oremos en el nombre del Padre, del Hijo y del Espíritu Santo. Dios Todopoderoso, te bendecimos por la grandeza de tus eternas misericordias. Te suplicamos nos bendigas con tu divina presencia en esta tarde. Dirige los trabajos que tenemos por delante de manera que redunden en beneficio de la audiencia presencial y virtual. Que la experiencia sea una de aprendizaje y crecimiento, tanto educativo como también profesional. Agradecido, Señor, por tu presencia y compañía en esta tarde. Hemos orado en el nombre del Padre, del Hijo y del Espíritu Santo. Amen.
You may be seated. It is a pleasure to present to you Dr. Juana Negrón Berríos, Chancellor of our campus, who will address us in a greeting.
Buenas tardes. Good afternoon to everyone. It's a pleasure to have you in Barranquitas campus for this special talk in the area of epigenetics. Also, not only the student and faculty in this amphitheater, but those who are through internet. I want to give a special welcome to Dr. Mary Gelling. It's a pleasure to have you here in Barranquitas, and especially in the fact that you will be explaining a relative new area in science, which is the area of epigenetics. We appreciate your presence here, and we expect to keep on with the collaboration that we have through Dr. Alok, MIT, and also through your lab. So thank you very much for being here in Barranquitas. This subject is very interesting, very important. I would like just to say a few words, especially for the students. Epigenetics, like I mentioned, we could say is new in many ways. It is an opportunity to understand a series of phenomena which occurs between the genetic makeup and the environment. It's a kind of changes in which other part of the structure of DNA, not only the nucleotide, can change. Important things like nutrition, our lifestyles in general, is directly linked to epigenetics. So there is a lot of opportunity in this area. Here in our campus, we have very small project related with epigenetics and plant, in particular, the impact of heavy metal on epigenetics of plants. This is also very important during these days in which we have the climate change. Plants, especially crop, have to adapt to those changes in the environment. So epigenetics, I'm pretty sure they plays a very important role in that sense. So I invite students to consider epigenetics, especially those in the master and PhD in biotechnology, as an area for research. It is pretty exciting, and there is a lot of things going on in this area. A small announcement. I imagine that you all know that we are in the process of reaccreditation. So that means next week we will have a committee from Middle State that will be here in our campus for at least 3 days, evaluating our campus, and I'm pretty sure that everything would be all right. So I will ask you to be available, especially the faculty, in case that this committee might have some question related, especially in the area of academic. So I hope that everybody will enjoy the presentation from Dr. Gehring, and a special welcome also from people from Guayama. I see some of them around here, and other campuses that come to Barranquitas campus. So I hope that you enjoy this afternoon here in Barranquitas campus.
We thank Dr. Negron for greeting us on this occasion. At this time, we invite our doctoral student, Gerardo Laureano Rosario, to introduce our guest speaker, Dr. Mary Gehring, who will be presenting on mechanisms to maintain plant epigenetic stability across generations.
Good afternoon. I would like to present our guest speaker, Dr. Mary Gehring. Dr. Gehring began her scientific career at Whelan College, where she studied biology as an undergraduate and began doing research on plants. She earned her doctorate in plant biology from the University of California, Berkeley in 2005 in the lab of Robert Fisher. Her postdoctoral research with Stephen Henikoff at the Fred Hutchinson Cancer Research Center in Seattle was focused on epigenomics. Gehring started her lab at Whitehead's Institute and MIT in 2010 and was named the Thomas D. and Virginia W. Cabot Career Development Professor by MIT in 2011. She has been awarded the Rosalind Franklin Young Investigator Award, a Spew Color Award, and other honors. In 2020, she was named the London T. Clay Career Development Chair at Whitehead's Institute. Her lab investigates epigenetics, mechanism of gene regulation in plants, and it's a great honor to welcome Dr. Mary Gehring.
Thank you for the kind introduction and for the words of the Chancellor and the Chaplain as well. It's great to be here today. I've just arrived in Puerto Rico yesterday, but I've had a wonderful time so far. And I want to thank you all for being here in person and also those of you that are watching online. Today I'm going to talk about plant epigenetics and some of the research that's been going on in my lab with regards to epigenetics. First, I want to start with some introductions of the term. What does epigenetics mean? And how does that compare to genetics? Probably you're all familiar with the idea of genetics, that we have certain traits that are controlled by DNA that are passed down from parent to progeny. And if there's changes in DNA sequence, that can cause a change in a trait or a phenotype, as we would say. So this is how we define genetic. Epigenetic is very similar with the addition of one word here. So epigenetic refers to heritable changes in traits that are not caused by a DNA sequence. And so you can have two organisms that behave quite differently or look quite differently, and you sequence the DNA of those organisms, and it's exactly the same. And so we're looking for something beyond the DNA sequence of A, T, C's, and G's that will explain a certain trait that an organism has. So here are a couple of classic examples of an epigenetic phenomenon, one in tomato and one in mice. So these mice here are siblings, and you can see they have a range of coat colors, from a very dark brown mouse to a lighter tan or blonde mouse. And the gene that is responsible for controlling coat color in mice is known. And so if you sequence that gene in these different sibling mice, you might expect that there would be a difference in the DNA sequence between this light-colored mouse and this dark-colored mouse. But that's not the case. The sequence of the gene is exactly identical among these siblings. So there's something else that must explain why these organisms look different. So the same here with these tomatoes. So here's a normal tomato that's ripening, and on the left is a tomato that never ripens. So this yellow tomato was found in a field, and they called it the colorless non-ripening mutant. So it would never turn red. But eventually they identified the gene that controls that ripening trait, and when they sequenced it, it had the exact same sequence of A, T, C's, and G's. Okay? So again, we know the genes that are responsible for these traits, but there's not any difference among these different individuals that look very different. And so we would call this epigenetic. Here's some other examples of epigenetic phenomenon in plants. On the right, these grapes, these red grapes versus the green grapes, look quite different. But actually, the color of the skin is due to an epigenetic difference. They have the same gene for making the purple pigment, but in these grapes, that gene is expressed, it's transcribed, but in these grapes, it is not expressed. And so there's some difference here, not at the DNA sequence level, that leads to this difference in fruit color. Here on the left, this is a field of winter wheat. And it's been well known that wheat, as well as many other species that are grown in the northern latitudes, actually rely on a prolonged period of cold in order to flower and then make seeds. And this period of cold is basically, the plant has a way of counting how many days it's been in the cold. And so in the northern latitudes, once a plant has been in the cold for about 40 plus days, then it should be spring, and so the plant knows to start flowering. So again, this is epigenetically controlled through DNA, how DNA is packaged. Okay. So why are we studying epigenetics in plants? Well, I've shown you a few examples of traits, skin color, flowering time, ripening, that I've told you are controlled epigenetically. And so we know here, this third point, that there are many agriculturally important traits that are influenced by epigenetics. And there's also interesting research, as was mentioned, in terms of how the environment, different conditions can affect this. Many of the epigenetic phenomena that we know about were actually first discovered in plants. So there was a lot of work in maize or corn in the 1920s through the 1940s, where many of these epigenetic phenomena that we study today in plants and animals were first described. So there's a really long history of studying epigenetics in plants. And also, so the way we study epigenetics is we still take a genetic approach in many cases where we're still mutating genes or we're working with genetic mutants to determine what the normal function of a gene is. And we know that in plants, there's some genes that control epigenetic traits where we can mutate the gene and the plant is still viable. It's not dead. And so because of that, there's some experiments we can do in plants that we can't do, for example, in mice, where if you mutated the same genes that control epigenetic inheritance in mice, the mice don't live. And then finally, one really interesting area of future research is whether we can actually use epigenetic engineering to create traits of interest in agriculturally important crops. And can we do that in an inducible way? So there's a lot of interest in understanding epigenetics enough such that we can manipulate it in crop species. So instead of creating a transgenic plant or a mutant plant that had a certain trait, could we change the DNA methylation state, and I'll talk about methylation in a minute, to change how a gene is expressed, which might then alter a phenotype. And so this is really an area of emerging interest in the field. Okay. So what are some of the mechanisms whereby you see these effects? So on this slide, I've highlighted a few of them. And as the Chancellor mentioned, a lot of this is how the DNA is packaged inside a nucleus and how it's modified. So the genome of an organism, so if you took our genome and you stretched it end to end, it will be several feet long. But that DNA has to fit inside a nucleus of a cell, which is very, very small, about 5 micrometers. And so there has to be an incredible amount of packaging of the genome in order for that to occur. And that packaging of the genome can really influence its function. It can influence how genes are expressed. Okay. So that's shown here. We start here from what we would call a naked DNA molecule. That molecule can be modified by chemical modifications called DNA methylation. And this is what I'm going to spend my entire talk focusing on. But I just wanted to mention some of these other mechanisms of epigenetics. We know that non-coding RNAs can influence the transcription of genes that they match or sequences that they match. My lab also works on those quite a bit. And then how DNA is wrapped around histones. So histones are the very basic proteins that are used to package DNA. And so across plants, animals, and other eukaryotic species, DNA is wrapped around these histones, and the tightness of that wrapping or how the histones themselves are chemically modified can affect gene function. Oops. Okay, so I'm going to focus on DNA methylation today, and specifically what I mean when I say DNA methylation is this base, 5-methylcytosine. So cytosine is one of the 4 bases in DNA, A, T, C, and G. But it can be modified where you can add a simple methyl group to that 5' carbon, and that makes 5-methylcytosine. So there's a class of enzymes that does this reaction. And so actually in our genomes, in many plant genomes, in most plant genomes, and in fungal species, many other kinds of species, you find significant levels of 5-methylcytosine in the DNA. Typically, this modification, we would call this an epigenetic modification, is associated with gene silencing. So if you have a DNA sequence where methylation has been added, that tends not to be expressed, meaning RNA isn't made and protein isn't made. And so we know that this modification is involved in several important processes in controlling transposable elements, in genomic imprinting, and in things like nuclear dominance. Okay. And so in fact, this first example I showed you of this tomato that never ripens, that phenotype is due to a DNA methylation difference between the wild type and the mutant plants. So there's this gene, CNR, which stands for colorless non-ripening, and it's expressed, it makes RNA, that gets translated into protein, and that protein is required for the fruit to turn red. But what happens what has happened in this mutant is there was a spontaneous change in DNA methylation. So throughout my talk, I'll indicate methylation with these circles. where that DNA methylation is in the regulatory promoter region of the gene, and it silences its transcription. So it doesn't make any RNA, doesn't make any protein. Okay. Furthermore, this epigenetic change, this difference in DNA methylation, even though it occurred spontaneously, it actually is then heritable. So there's a mechanism for copying this information from one cell division to the next and one generation to the next. And so they reported in this paper that they looked after several generations in a field of tomatoes to see whether they were all still yellow. And in fact, they were, except for 3 individual fruits. So they found 3 fruits with a little bit of a red stripe. And so what we think happened there is that likely DNA methylation was just lost at that gene in those particular cells. Okay. So once a methylation change occurs, it can actually be heritable and create this phenotype that behaves in many ways like a genetic trait. It's heritable from parent to progeny, but then when you go in and look at the sequence of the gene, there's no difference. And so why is there this heritability of methylation? I just want to run through the mechanics of this with you a little bit. So methylation in plant genomes is most frequently found in what we call the CG dinucleotide context, where if you're reading the DNA from 5' to 3', you have a methyl C, and they're standing for methyl, followed by a G. And then on the opposite strand, you also have, if you read the DNA in the same direction, a methyl C followed by a G. So this is a symmetric DNA methylation. You can also find in plant genomes, this is in contrast to animal genomes, methylcytosine outside of this CG context. So you might find, for example, a methylated CCT. Okay. After DNA replication, the new strand of DNA that's made won't have any methyl marks. So the methyl group is added after replication. Okay? So you have a new strand of DNA with no methylation. But there's an enzyme that's called a maintenance methyltransferase that actually moves along with the replication fork and deposits methylation on the new strand of DNA based on the pattern of methylation on the parent strand. So it uses this as a template to know where to put methylation on this strand. And so that works very well for these symmetrically methylated cytosines, these CG sites. And then there's additional mechanisms that are required to maintain this asymmetric methylation here, which you can see after DNA replication. There's no evidence, right, that it was ever present there. And so there's a de novo methyltransferase system. This is just a slightly different methyltransferase enzyme that is guided to targets by small non-coding RNAs to methylate this new strand. So this is a process for these asymmetric methylcytosines that has to happen repeatedly with every round of DNA replication. Okay. And so in my lab, we're primarily looking at epigenetics in this system, in Arabidopsis thaliana, which is a plant model system. It's in the mustard family. And the reason we're doing that is it's very, very easy to work with. So it's like working with fruit flies or lab mice. It has a very small genome. It has a pretty short life cycle for a plant. So it's about 2 months from seed to seed. And you can do a lot of genetic crosses with this species, Arabidopsis, very easily. We can use CRISPR mutagenesis to make mutants. And there's worldwide genetic and epigenetic diversity. Okay. And we can use this plant. This is a technique I'll go into in some detail to actually measure DNA methylation at very, very high resolution. So we have techniques where we can say at an individual cytosine in a genome that's 150 million bases, we can say, okay, at this particular cytosine, on this particular chromosome, this is the methylation state. And one of the ways we do that is through a sequencing method called sodium bisulfite sequencing. So let me just walk you through this. Let's say this is your strand of DNA, and there's a methylated cytosine here and some unmethylated cytosines. If you take isolated genomic DNA and you treat it with this chemical called sodium bisulfite, which is a very easily attainable chemical, it reacts differently with unmethylated and methylated cytosine. So it reacts with unmethylated cytosine to form uracil. And then it doesn't interact with methylated cytosine. So if you take this DNA and you sequence it, the methyl cytosine will be read as a cytosine, and the unmethylated cytosine will be read as a thymine, because U will be replaced with T in the DNA. And so now you have your sequence of your sodium bisulfite treated DNA. You can compare it to the reference sequence and then see, okay, where is there a T where I expected a C, and where is there a C where I expected a C? Okay? And you can do this at the level of individual genes, individual sites in the genome, or you can do it for the entire genome at once. And so if you do it for the entire genome, this is what you see for the Arabidopsis genome in terms of where methylation is. So the Arabidopsis genome has 5 chromosomes, which are laid out here. And this is the methylation level from sort of 0% to 100%. And then I've also plotted here the distribution of genes in the genome and the distribution of transposable elements. And I'll explain what those are in a minute. What you can see here is you see these humps of methylation sort of in the middle of each chromosome. This corresponds to where the centromere is, and also where you have very high levels of DNA transposable elements. So let's zoom in here. So here is a region of the genome where there's no genes, it's just transposable elements, and there's very high DNA methylation in different sequence contexts. So here these H is any base other than G. This type of methylation is very static. So you'll find this, if I took 100 different Arabidopsis plants and I looked at their methylation, they would all have high methylation of these sequences. Okay, so what are these transposable elements? Transposable elements are selfish pieces of DNA that simply exist to increase in copy number. They don't really do anything for the organism. They basically just encode proteins that allow them to copy themselves. And so they're sometimes referred to as junk DNA, but they're actually a huge fraction of our genomes. Okay? So in our genome, about 44% is transposable elements. And in plants, you can have even higher numbers. So in corn, about 85% of the genome is this transposable element DNA. So these transposable elements, as I said, they can move around and increase in copy number. And so to prevent that, organisms have evolved what we call genome defense mechanisms, a way to stop that transcription of the transposable element and movement. And this is largely through DNA methylation. And so that's what I was just showing you, where those transposable elements are very highly methylated. And what that does is it stops them from being transcribed so that they can't move around the genome. Okay. So that's a major feature of really most eukaryotic genomes. If we look at some other regions here, what we can see if we go in a gene-rich region is there's very little methylation for the most part. There's some CG methylation. But the regions of the genome that I find most interesting are like this. So this is a region with several different genes here, but there's also a transposable element, a small transposable element fragment. Excuse me, sorry, that was really loud. And this transposable element is very heavily methylated. And it turns out that these are the regions of the genome where we see the most dynamic changes in DNA methylation. At these transposable elements that are living near genes. Okay. And so these patterns that we see in the genome, all of these patterns are created both by active methylation, which I told you a little bit already, and also active demethylation. So there's a mechanism to add methylation to cytosine, and there's also a mechanism to remove it. And it's very important, as I'll describe today, it's very important to have a balance between these two opposing activities, adding methylation and removing methylation. And so in general, methylation promotes silencing, and demethylation promotes gene expression. So these enzymes that remove DNA, they actually don't directly remove the methyl group from the cytosine. What they do is that they remove the entire methylcytosine base through a DNA repair process called base excision repair. And I'll just very quickly walk you through this. The details aren't too important for this talk, but basically these enzymes recognize methylcytosine. They excise, they flip the base out of the double helix, and then it gets cleaved off. That leaves an empty site in the DNA here that is then repaired by DNA polymerase and a DNA ligase that adds cytosine back. So through this sort of complicated process, you have replacement of methylcytosine with cytosine. And so what I want to go into more in depth today is two aspects of my research program. One is, how do you actually maintain these patterns of DNA methylation from one generation to the next? I didn't put in a slide showing this, but if, for example, if you lost all methylation in the genome, that's typically not a good situation for a plant. The plant will grow slowly. It will not produce very many progeny. And also, conversely, if you hypermethylate the DNA, that also is not good. So I'll talk about how you maintain this fidelity and then the targets and function of DNA demethylation in plants. So zooming in on this is a genomic region similar to what I showed you before, where you have a gene here. This is the start of the gene. And then nearby you have some transposable elements. And you can see that there's DNA methylation in that transposable element, presumably silencing it. But there's sort of a challenge for the genome when you have genes next to transposable elements, because there could be spreading of that methylation such that it silences a gene. And so it seems that one really important function of this demethylation process is to stop that spreading. And we know that because if we disrupt DNA methylation, this isn't a mutant of the DNA demethylase gene, so we've mutated the demethylase gene. That means we can no longer remove those methylcytosines from the genome. And what you can see is that there is now methylation spreading from this transposable element into the gene. And that actually can then silence that gene inappropriately. And so there's this challenge of how do you maintain the genome in this state where transposable elements are methylated and silent, but genes are kept relatively free of methylation and can be expressed. It's sort of the type of balance that you also think about. We call this a genome defense mechanism against transposable elements. But there's many cases where, for example, with our own immune system, we want it to attack pathogens, but we don't want it to attack us. And so there's always this sort of fine line in these systems of how do you maintain this homeostasis, okay? So what we found, so the point there just being that you have this demethylation pathway to protect genes from this methylation in TEs that could spread. And so I'm going to show you what we've discovered about how you actually balance the addition of methylation and the removal of methylation so that the genome is kept in this state where genes are expressed and transposable elements are epigenetically silent. Okay. And this started with an observation we made. We were looking at expression of the DNA demethylase gene, which is called ROS1. And we found that the expression of the gene encoding the demethylase was altered in mutants that affected DNA methylation. So this is just looking at, this is some qRT-PCR data looking at different mutants here on the bottom that affect the maintenance of methylation and compared to wild type. So in wild type, we have an expression level of ROS1 that we set at 1. But then in all these mutants, we found about 10-fold reduced expression. And this was very curious that you disrupt a methylation pathway, and it's the demethylase expression is affected. And so we want to understand why that was. We could also see this at the protein level, not just at the transcript level. So this is a reporter gene we made where we took the ROS1 gene and we fused it to a luciferase reporter. Luciferase will give you this a nice chemiluminescent reaction if the gene is being expressed. And so here is where this transgene is expressed in a normal wild-type plant. These are the leaves. This is the center of the plant where the meristem is. But then we found in a methylation mutant, which is called RDR2, that there's much less expression. So there's less RNA and there's less protein. Okay. So when we looked at the ROS1 gene, what we found is that it is also DNA methylated. And actually, it's one of these interesting genes where there's a transposable element very close to the gene. Okay? So this is just showing you the structure of the gene with the exons and the introns and where it's transcribed from. And upstream, there's a very short transposable element. And in this region, there's DNA methylation, both CG and non-CG methylation, okay, which is indicative of a very active methylation process. And so normally, if you saw a gene like this and you saw methylation in the promoter, the prediction would be that that methylation is silencing for the gene, that it should be turning it off. But we found when we disrupted DNA methylation, so in those mutants, we found that we lost methylation at the gene, and its expression was actually going down. So that's opposite to what I told you we typically expect to happen. Okay? And so this was the big surprise, that at this gene in particular, in contrast to most other genes in the genome, this DNA methylation seems to be actually promoting gene expression. And we could show that through, I'm not going to go in detail the experiments that show that, but we could manipulate the methylation there, we could target it, we could remove it, and we could show that those specific methylcytosines were actually regulating ROS1 expression. And so what we find is that at this gene that encodes a demethylase, you have an active methylation pathway that's adding methylcytosine. And this is increasing transcription. But we also found that the ROS1 protein, the demethylase, targets this same region. It targets its own DNA locus in order to demethylate it. Okay? So ROS1 is removing methylcytosine there. And so you have both the methylation and the demethylation pathway converging on the demethylase gene. And again, here the surprise is that removal of methylation is actually decreasing transcription. Okay? So this convergence of methylation and demethylation at the same locus is not really that surprising. But what is really surprising is that it has this opposite effect on transcription. So these expression outcomes are opposite to what we see at typical genes. And so what this led us to conclude is that we think that this mechanism, that this regulation of the demethylase by DNA methylation and demethylation is actually important for maintaining the balance of those activities in the genome. Okay? Just to run through the model, we have what we would call a negative feedback loop here. And it creates what we're calling an epigenetic rheostat. Okay? So at a typical gene, if there was methylation in the promoter, that would typically be repressive. And if demethylation was acting there, that would be activating. Okay? The opposite at ROS1. So let's imagine if you had increased methyltransferase activity in the genome, you would have increased DNA methylation at ROS1, which would actually lead to transcription of the gene. So you would increase demethylase activity. And the opposite is also true. So you have this mechanism, this rheostat, where you can dial up and dial down the level of demethylation activity in the genome just by the fact that these pathways are converging on this gene locus, but doing so in an opposite manner. So our idea was that probably this is going to be important for maintaining this balance of DNA methylation in the genome. And so we created some plants. I won't go through the details here, but so I'm referring to this circuit as an epigenetic rheostat. We created plants where we disrupted that rheostat and looked to see if they had a phenotype. And in fact, so this is a normal wild-type Arabidopsis. What we found in these plants where we disrupted the rheostat is their leaves are curled. And this is actually due to a change in DNA methylation at a locus that controls leaf shape. And interestingly, what we saw is that this phenotype worsened over generations. So when we disrupted the ROS1 regulatory circuit, we would see this curly leaf phenotype just in a few plants, maybe 5%. But then over time, from generation to generation, this phenotype would increase. And so what that shows us is that this ROS1 rheostat, or this regulatory circuit, is really important for maintaining leaves, maintaining plants in this normal wild-type state. Okay? So we think this rheostat is really functionally important, and we want to understand much more about, well, how does DNA methylation actually promote transcription? Oh, thank you. That's very kind. How can we actually, why would DNA methylation actually be increasing expression? Now, we haven't figured that out yet, but this is a project that a graduate student in the lab is working on. So I'll just mention a little bit here. So what we found using some CRISPR genome editing, we started removing different pieces of DNA at the ROS1 locus and seeing whether or not its expression was still sensitive to DNA methylation. And so what we did here is we removed a short 800 base pair region, 5' of the gene, and what we found is that it was no longer methylation sensitive. So this took out most, but not all, of the DNA methylation, and most, but not all, of that transposable element that's present in the 5' region. And what we found is that, so in wild-type plants here, you have in white, ROS1 is expressed at a certain level. That's reduced in a methyltransferase mutant, DRM2. But then if we delete this 800 base pair region, what you can see is that expression is increased in wild-type, and expression is increased in the mutant. I'm just going to take a drink of water. Hold on. And so what that means is that normally this is acting as a silencer. Okay, so let me show you this in cartoon form. This means that normally this region that it's methylated, when it's methylated, it's not as good of a silencer as when it loses methylation, okay? So when it's in its relatively hypomethylated state, for example, when it's been demethylated, it is acting as a silencer to the nearby gene. But when it's methylated, it can no longer act as a silencer. So we still don't know why this is, but some of the models we're exploring is, well, maybe there's a transcription factor or a transcriptional repressor that can bind when this is unmethylated, that represses ROS1 transcription, but perhaps when it's methylated, now that transcriptional repressor can no longer bind. And so we're screening for some proteins to see whether or not they bind to that piece of DNA differentially depending whether or not it's methylated. And so we have a few candidates for that right now. Okay. And so in this first part of the talk, the majority of the talk, what I hope I've shown you is that you really need this balance between methylation activities in order to maintain epigenetic fidelity. And so when we disrupted that by disrupting the rheostat, what we saw were those plants with curly leaves, and they also had some other phenotypes as well. That it's the DNA demethylation, the coordinate regulation of demethylation by ROS1 is really regulated with de novo methylation, and that can maintain stability. And I think this concept that you have to have a balance between methylation and demethylation, we're calling that epigenetic homeostasis, doesn't apply to plants, but many other organisms as well. So I just wanted to mention one example of that in mammals, and that's related to cancer. So in our genomes, similar to the Arabidopsis genome, most of the methylation is found in transposable elements, and genes are kept relatively free of methylation, and they can be expressed. But one of the key hallmarks, one of the first things that goes wrong in cancerous cells is that actually those transposable elements lose methylation and genes gain methylation. Okay? And so what you find in cancer cells is that you can detect expression of these transposable elements and also silencing of genes that would normally help prevent cancer, tumor suppressor genes. So if you silence a tumor suppressor gene, now cells can divide in an uncontrolled way. And interestingly, some methylation inhibitors, like azacitidine, which I was talking to Gerardo about, are actually used to treat cancers. So I think this concept of epigenetic homeostasis that we've uncovered in Arabidopsis in a plant is also useful for thinking really across systems as well. Okay. So now I want to transition to the second question I posed, which is where does this demethylation actually occur? I told you a lot about where methylation occurs, but I want to focus on some of the processes controlled by DNA demethylation. So there are 4 of these demethylases in the Arabidopsis genome. So I told you about ROS1 and how it's regulated. But there's 3 other demethylases. They're called DML2, DML3, and DME. So these 3 genes, you can disrupt them, and they're not essential. So the plants have some phenotypes, but they still make seeds, and you can get viable plants. This gene, DME here, is an absolutely essential gene. So this demethylase is expressed during reproductive development. And if you mutate it, you don't get viable seeds. So these are normal wild-type Arabidopsis seeds. If you mutate this DME, you get these seeds that are very large, but eventually they collapse. Okay. And so for a long time, we haven't been able to study genetically the function of these DNA demethylases because we couldn't make a mutant in DME that would be transmitted to the next generation. But we did some genetic tricks very recently, and we were able to create plants that in their vegetative or somatic tissues lack all 4 of these DNA demethylases. And I'll refer to those as DRDD mutants. And what we found, so we took these plants and we looked at gene expression by whole genome RNA sequencing, and we also looked at DNA methylation by whole genome bisulfite sequencing, and then another method called enzymatic methyl-C. And we did this from the same leaf so that we could directly compare those methylation patterns with gene expression. So you look at DNA methylation, what we found, so here's a wild-type methylation pattern upstream of a gene, very little methylation. We find if we mutate ROS1, DML2, DML3, you start to see a little bit of hypermethylation there. But then if you mutate all 4, you get this extensive hypermethylation, and that can affect the downstream gene. So we identified several thousand of these sites in the genome. But one thing that we noticed when we were growing these plants for the first time is that they flowered early. So here's wild-type plants, DME mutants, ROS1, DML2, DML3 mutants, and then here's a mutant where we don't have any demethylase activity, and you can see it's flowering, whereas the plants next to it are not. Okay. And so we were really interested in understanding, well, what's the connection between demethylation and flowering? Okay. So flowering is when a plant flowers is pretty much the most important decision a plant ever makes. Because it's deciding when it's going to make seeds. And so that has to be done at a certain time. You don't want a plant making seeds and flowering when it's freezing cold outside. And so there's a lot of inputs into flowering time, cold, day length, the type of light, the temperature, how much sugar is available, osmolarity. And so there's many, many inputs into this pathway. But one of the key ones is a gene called FLC. And we found that FLC has really, really reduced expression in these demethylation mutants. Okay. So what's FLC? FLC is actually a repressor of flowering. So when the gene is expressed, that keeps the plant from flowering. So high levels of FLC delay the transition to flowering. Eventually that can be overcome by some of these other pathways, but it really delays it. And so some of the ways that FLC expression is overcome is through this exposure to prolonged cold that I mentioned with winter wheat in the beginning. So the plant uses different chromatin modifications at FLC, histone modifications, in order to silence it as the plant experiences more days of cold. And also just age of the plant. So as the plant gets older, FLC expression reduces. And so if you reduce FLC, you get rid of this block, and then the plant can flower. So it turns out that FLC is the target for demethylation. But we never realized it before because in wild type, there's basically no methylation there. So this is the FLC 5' region. This is the upstream region. It's just the gene's pointing this way in the genome, so it's sort of backwards here. And there's this region where there's some repeats and a little bit of methylation there in wild type, but really nothing. What we found in the DRDD mutant is that you have this hypermethylation, and that hypermethylation is correlated with that low level of FLC expression we see in the demethylase mutant. What's really interesting about this is that, I haven't gone into this in detail, but we know that when we see small non-coding RNAs in a region, that means that active methylation is occurring. And we see that in wild-type plants, but there's no methylation there. And so what that suggests is that at this locus, you have methylation being added, but immediately removed. Okay? We don't really understand why that is. It seems like a futile cycle. Why target methylation to a place simply to have it removed by the demethylases? But we think that perhaps there's a certain point in development where it is important for this gene to be methylated. And so what we think is happening in these mutants is that this gene is getting methylated, and that is what's leading to its transcriptional downregulation and thus the early flowering. Now, this is looking at a mutant context, but we really want to understand, well, is there any role for methylation of this gene simply in a normal wild-type developmental context? And for that, we turned to the fact that Arabidopsis is spread throughout the world, and there's a lot of genetic and epigenetic variation there. Okay, so this is a map of where you can find Arabidopsis thaliana. I don't see a red dot down here, but maybe you guys should keep a lookout when you're walking around. It would be nice to have a collection from here. But you can find it, in the Cape Verde Islands, for example, the Canary Islands. And so we wanted to look and see for Arabidopsis that has difference in flowering time naturally, is there any connection to DNA methylation? And so what we did, so there's about 1,000 strains that have been sequenced, and also their methylation, their epigenome has also been sequenced. And so we have DNA sequence data, DNA methylation data, and we have flowering time data. And what we did was we found very genetically closely related Arabidopsis that had a difference in DNA methylation at the FLC locus, and we wanted to see whether or not there was a difference in flowering time. So we're trying to control for genetic variation and just look at epigenetic variation. And in fact, what we found, so we were looking at 14 different pairs of genetically similar Arabidopsis that come from different regions of the world. And if you compare strains that have high methylation to low methylation, what we see is this difference in average flowering time for 12 of these 14 different pairs that we were comparing. So that basically when you have this difference in DNA methylation, that can give you a difference in 20 to 40 days of flowering time. So that's a really, really large difference. And now what we're trying to do in my lab is actually do some epigenetic engineering at the FLC locus, where we take one of these strains that has high levels of methylation and remove that methylation and see if that's sufficient to change flowering time. Or we take a strain that has very low level methylation, add methylation in and see if that is sufficient to make flowering time earlier. Okay. And so just to conclude this section, what we found is that in wild-type plants, this DNA demethylation process is really important for promoting FLC expression, which delays flowering. But I think more interestingly that there may be natural variation that exists within population in terms of where demethylation acts and also where methylation acts that can lead to these natural differences in phenotypes. And so we've shown that natural variation in the level of demethylation at FLC contributes to differences in flowering time that you see around the world. We're also looking at some other traits that might be impacted. We've found some additional phenotypes in these plants where they don't, the pollen dehiscence is not normal, so the pollen stays inside the anther and isn't released. And we're trying to understand the epigenetic basis of that phenotype as well as a few others. One thing we really want to know, and we don't know yet, is really where is it important during development that demethylation occurs? Is it important in all the cells? Is it important in just the embryo or in the meristems? And I think that's going to be really exciting for the future. So I'll end there, and I want to thank the people in my lab that contribute to this work. So this is my lab at the Whitehead Institute, a mix of PhD students and postdocs. We also have a couple of undergraduates in the lab. Most of this work I told you about was done by Ben Williams, who's now a professor at University of California, Berkeley, and a former graduate student, Deborah Pullman, along with Lindsay Betchen, shown here, who's a technician in the lab. I'd also like to thank the funding I have for this work, primarily from the National Institutes of Health, and also thank all of the cores at Whitehead, the research cores that are really important for our work, such as the Genome Technology Core, where we do all of our DNA sequencing. Thank you.
Thank Dr. Gehring for such an excellent presentation. We will now proceed to the Q&A session. To make your question, please raise your hand, and one of the technicians will hand you a microphone. Also, virtual guests can write their questions in the chat.
Very interesting talk. Is there any correlation between the amount of transposable elements present in a genome and the level of methylation that you get?
Yes, definitely. So if you did like an HPLC experiment to see the amount of 5-methylcytosine, there's much more in corn than there is in Arabidopsis, for example, and that's directly connected to transposable elements.
Yeah. My other question is, you mentioned that there was a transposable element that was present next to the ROS gene that was small in size. I was just wondering if the size of the transposable element is larger, would the expression pattern of the gene would be affected by the size of the transposable elements lying next to it?
Yeah, I think it would be. The transposable element next to ROS1 is actually really small. It's about 150 base pairs. So it's actually a remnant of a transposable element. It doesn't have any ability to move throughout the genome anymore, but it's still recognized as repetitive by the genome and is targeted for methylation. So I think you've hit on an important point that if it was like a really big transposable element, that would probably be silencing for ROS1. But there's something about this short region, which actually has a couple transcription factor binding sites in it, we've now found, that I think makes it regulatory in this way that I've described. Yeah.
I have one last question. So at the end you mentioned it's unclear if the methylation happens at the tissue level or the cellular level.
Yeah.
So how are you going to study that?
Well, we're starting to do some, so Elizabeth there on the left, she's starting to try and sort different populations of cells based on some markers we have. So we have some genes that are expressed in stem cells, and so you could sort those out and look at methylation specifically in certain cell types. We also have some reporter genes for methylation where we have a gene fused to GFP, and we know that when that gene is methylated, the GFP is off, and when the gene isn't methylated, the GFP is on. And so we can use fluorescent activated sorting to take GFP plus cells from GFP minus cells and then use that to see what the rest of the genome looks like in those cells. So we're using that GFP activity actually as a marker of demethylation activity.
A great presentation. In this area, this methylation process is not black and white. You know, how far do you see the possibility of using this knowledge of technology, let's say, to identify important crops, maybe from the nutritional point of view or commercial value in the market?
Yeah, I think, you know, for something to have commercial value, a sort of epigenetic engineering system, it would have to be pretty precise. You know, something I've been thinking about is what if you could design a system where it was responsive to in certain environmental conditions? So like when it was dry, you would have the expression of a gene that would help the plant conserve water. But you wouldn't want that normally to be expressed in rainy conditions because typically these sorts, you know, that there's a cost to the plant of expressing it. So if you could have like a methylation system where you change the expression of a gene rather than mutating it like we would currently do in breeding, then you could hopefully develop like an inducible on-off state. So we need both a way to direct methylation to a certain sequence, which I do think we actually have now with some of our knowledge about small RNAs and some of the CRISPR engineering we can do. We can direct methylation to certain sequences. But we also need to be able to direct demethylation to certain sequences and do that in an inducible way, like under a certain environmental condition, this system will be active and you'll add methylation or you remove methylation from a gene that would be important. Does that answer your question?
Yes. Okay. Because, for example, for cancer, many people are trying to identify molecular markers of methylation for certain type of cancer.
Yeah. Oh, so you're asking, maybe I misunderstood your question. You were asking.
That's hard.
Yeah, people are, I mean, there was a paper that just came out yesterday from a colleague of mine at the University of Georgia, actually looking at, you know, there's some crops like rice, for example, where there's many varieties and there's genetically not much distance between these varieties, but they may have very different phenotypes. And so he had a paper, I forget, it might have been in soybean actually, where you could actually use DNA methylation differences as a marker for evolution, so how far apart these were evolutionarily. So even if there wasn't enough time to be many DNA sequence differences between a couple of lines of soybean, you could see DNA methylation differences. And then some of those methylation differences might be important for different phenotypes in crop varieties.
We have a question in the chat.
Yeah, this is a really important question. And something that plant biologists have been thinking about for a long time. So is there a memory of an environmental stress, for example, that's epigenetically encoded? Elizabeth and I just wrote a review article on this that's just come out. And the evidence so far is limited. So people have looked to see, for example, if you expose Arabidopsis to drought conditions, what are the methylation changes, and do you see then greater resistance to drought in subsequent generations and corresponding methylation changes? There is a little bit of that, but it typically, so far what has been found is, like in the example of drought, is that there will be an effect for like 2 generations, but not a really long-term effect. And I think that sort of makes sense, that if a plant is exposed to a stress, it may be beneficial for its progeny to be more resistant to that stress. But over time, conditions can change. Certainly sort of weather conditions are quite variable. And so it may not make sense evolutionarily to have a really long memory of an environmental stress. But this is definitely something where there's still a lot of unknowns and a lot of active research in this area.
Hi, so wonderful talk. I had a question involved when you mentioned de novo methyltransferases. I just wanted to know if there been any research done on determining like the frequency of amount of the frequency of how much de novo methyltransferase happens in comparison to the other methods.
Sorry, say the last. So the frequency of how much de novo methylation happens
occurs in contrast to the
demethylation.
Yeah.
I would say probably there's actually more methylation activity than demethylation activity. So the de novo methylation, it's really interesting. I didn't go into the mechanism very much, but basically there's this whole machinery where small RNAs are produced, and those are actually guiding the de novo methyltransferase to where it should add methylation. And those small RNAs are primarily coming from transposable elements. So, it's something that has to happen every cell division cycle. Whereas the demethylation, I think of more as kind of cleaning up after the methylation system. So there's all this methylation that has to be put in place on transposable elements. And then the demethylations are going and pruning that methylation away when it gets too close to genes, for example.
Hi. Right here.
Oh, hi.
So besides methylation, have you worked with other types of post-translational modifications to study the expressions of FLC?
Yeah, so FLC is also, this is not work from my lab, but from other labs. We know that it's regulated by the polycomb group complex, which is an enzymatic complex that methylates histones, histone H3 on lysine 27. And so there's a lot of really beautiful work, for example, from Carolyn Dean's lab in the UK, showing that that cold responsiveness that FLC has, so its level decreases in the cold, is because you have a histone modification that occurs in the gene a bit downstream from where we found methylation. And then that spreads along the locus and silences it. And there's also some, it's a pretty complicated locus. There's also some non-coding RNAs that are involved as well. So that is actually the interplay between DNA methylation and histone modifications is really interesting. That's something we're working on in other projects in my lab, in our projects on seed development, where sometimes it seems that DNA methylation and the histone modifications are synergistic, and sometimes they appear to be antagonistic, and we don't know quite why yet. But it's certainly true that they're like all of these epigenetic pathways tend to feed into one another, and there's a lot of actually positive synergy in the silencing pathways. So where you find DNA methylation, you often find histone H3 lysine 9 methylation, and there's enzymes that recognize both those modifications. Yes.
So thank you for that presentation. My question is, you mentioned earlier about the histones and the RNA, interfering RNA.
Mm-hmm.
In your opinion, for epigenetic engineering, which one would you think would be more effective out of all three?
Yeah, I think DNA methylation is going to be the most straightforward for epigenetic engineering, because it is it's heritable in a way that histone modifications aren't. So, you know, as the DNA is replicated, histones are, there is some histones will be retained on the DNA, but there's not this sort of copying mechanism like there is for DNA methylation. So I think that's probably going to be the most effective strategy. But also tied up with the DNA methylation are the small RNAs. So like in my lab, when we want to direct DNA methylation to a locus, what we do is we make a transgene that'll express a small RNA that matches that sequence, and that's sufficient to target methylation. So you can have put in a small RNA to whatever you want, and then take advantage of the fact that the plant already knows once it sees a small RNA what to do with it, and it brings the methyltransferase there.
Thank you.
I have a question.
Mm-hmm.
Hi.
Hi.
Thank you for your speak. This is for my notes because I was taking it and something didn't click. You said that the wild type, they don't demethylize, or they do the metallization.
Oh, well, wild type, did you mean maybe at FLC specifically? But so wild type plants will do both methylation and demethylation. And then we were working with some mutants where we've disrupted the demethylation pathway. In the first part, when I was talking about, I left some things kind of vague because I didn't want to get too deep into the details. But when I was talking about those plants where we found curly leaves, there we had disrupted the regulatory circuit at ROS1, so we had disrupted the methylation and demethylation pathways both acting there. But if you have something more specific, you can maybe come up later and ask me.
Yeah. Thank you so much.
Okay, if we don't have any more questions, thank you again for this presentation, for giving us this important information, especially in the natural sciences field. We would like to excuse Dr. Alok Arun, Director of the NSF HSI STEM project, who, due to health situation, will not be able to moderate our next session titled In Conversation with Dr. Mary Gehring. We invite our guests to participate by sharing your question with us. We'll have some chairs so we could do the talk.
Okay. Nice to see you. Thank you for moderating.
So we have a few questions.
Okay.
First of all, who or what inspired you to pursue a career in science?
I think when I was in high school, I had a very good biology teacher who also happened to be my dad. My dad was my high school biology teacher. But he was very good, even though he was my dad. And, you know, so when I was in high school, I started to learn about molecular biology. So he had taken it upon himself to try and understand molecular biology and how do you like isolate DNA and run gels. And so I started to get a little taste of laboratory science when I was a high school student in northern Michigan. And so then I was, you know, I was always very curious about science. I wasn't really sure when I started undergrad, but doing undergraduate research really made me want to continue in science. I really like being in the lab and doing things with my hands, but also thinking about experiments.
So we have a question about that, so I'm going to ask right away. What is the importance of students participating in research program during their undergrad and graduate studies?
Oh yeah, I think, I mean, it's essential really to being a scientist. And I think, getting that taste of research in the laboratory is what gets students excited. So I think as much as undergraduates can have experience in the lab, either through a class or an independent project, is really, really important. I think what you often don't realize when you're just taking classes is how much is unknown. You can read a textbook or hear a lecture and think, well, we understand everything. But then when you're in the lab and doing experiments, you realize, wow, there are all these problems out there that nobody knows the answer to, and maybe I'll be the one that figures out the answer.
Great. What challenges did you face during your academic career, and how do you overcome them?
I think sort of related to what I just said, challenges in an academic career. I mean, of course, there's a lot, you have to do well in your courses and things like that, and study a lot. But I think challenges is, like with science in particular, with research, is that there isn't an answer. And most of our hypotheses that we make as scientists will be wrong. So most of the things I think about DNA methylation right now, many of the answers I told you, in 20 years they may be shown that they're wrong. And so the fact that science is at the frontiers of knowledge is very exciting, but also very challenging. So you have to, I would say, a big academic challenge is becoming comfortable with the fact that there may not be a right answer to something, and that you have to really figure it out.
Good. What advice would you give to a student interested in a similar career path?
Yeah, I think if you're interested in, so I took a pretty straight trajectory from undergrad where I did research as an undergraduate. I wasn't sure if I wanted to continue with a PhD, so I did work in consulting for a year, but then I quickly decided I would prefer to be in a lab. So I think really the key thing is, if you want to follow that path to eventually doing research and being faculty, or doing research and working at a company, is really starting to get actual exposure to a laboratory environment. Even if it's through taking a lot of lab classes as an undergrad, but that's where you have to start.
What is the importance of mentors, and how did your mentor help you in your academic career?
Oh, mentors are key, because in terms of an academic career, there's so many things you don't know. There's a lot of assumed knowledge, I would say, where, oh, well, of course you understand what it's like to be a graduate student. But many of us who don't have anyone in our family who's ever been involved in science, it's really kind of mysterious what those different steps involve. And so mentors, I think, are critically important to serve as a guide. They can't answer all your questions, but they can serve as a guide. They can introduce you to people, and they can really help you think about, okay, what's an interesting question to be asking? What's an interesting problem to be working on? Even now, the mentors that I had, like my postdoc mentor, is really important to me now, even though I've been out of his lab for 13 years, but he's someone in the field who sort of has my back, and if there's opportunities that come along, he'll suggest me for them, and so it's really important to find those people that can be your champions in your chosen field.
Great. You're here because of networking. How is important networking, and what is the best way to connect with students and professionals in the field?
Networking is really hard. For me, as someone who is naturally shy, it's not something that I like to do, to just talk to strangers. But it is really important to know people, and a lot of how you get jobs, for example, and my husband's in biotech, and a lot of that is knowing someone at a company and they need a position. And I think one way to do it is first you sort of start with your peer group. You start with other students, graduate students, and then you sort of try and build from there. I think your mentor is really important for that. You know, one thing that a mentor should be doing is introducing you to other people. Also going to conferences in your field is really great for that. It can often like feel awkward to, you know, introduce yourself to someone. But at a conference, that is expected. And so if you can go to a conference in your field, you can meet those people that are really important in that career path. And they, you know, many people want to help the younger generation come up.
Great. What is the most rewarding and most challenging part of your job?
Most rewarding. Most rewarding is still discovering things. So right now, you know, I'm not doing the experiments in the lab myself most of the time, but you know, when somebody in my lab tells me about a great result, that's the most rewarding. Or when you see a student really switch kind of from being student to sort of the expert in the subject, that's really exciting. Toughest part is always finding grant funding for research. That's always a challenge. And there you have to get very used to rejection as well. So writing grants, I would say, is the toughest part.
You have contributed to over 40 publications. Congrats.
Thank you.
Can you share your insights on writing scientific papers? How have you contributed to your career?
Yeah, writing papers is really important. You know, my perspective is papers are the way we share the knowledge that we've gained. And so even if you've done beautiful experiments in the lab, if you don't write a paper and share them with the world, it's really not important. So you have to, you know, it's really something we have to do. I mean, writing a paper is always a very long process, and I think it's something that you get better with time. And so that's one of the things that I really try and mentor my students on is how to write scientifically. It's quite different from writing, you know, in prose for your English class. The most important thing is, I think, to be as logical as possible in your writing and think, how can I guide the reader through what I want them to know about? Sometimes that may be writing a paper as the experiments actually happen, like the order in which you did them. But most oftentimes, it's better to write a scientific paper in terms of what we say is telling the best story. So even if you learn this and then this, what is actually the way you can put those results together such that the person who's reading the paper really understands the question you had, the approach you took, and then what you found.
Exactly. Then what is the point to publish something that the reader won't understand? Right.
It's really, really important to, I mean, we spend probably writing the paper is, you know, as important as doing the experiments.
Of course. And that's the way to share the information.
Yeah.
The new knowledge.
Yeah, exactly.
What are the things you consider before conducting a research project?
What do I consider before conducting a research project? You know, is it an interesting question? Is it something that hasn't been addressed before? Sometimes I'll have a great idea, but then I go to the literature and like, oh, someone did this in 1985. So it's also important, in addition to writing papers, really reading a lot of papers. So it is an interesting question, and it sometimes depends on who is doing the work. So if it's an undergraduate versus a graduate student, it's going to be, you know, the approaches we're going to take are different. An undergraduate is going to have less time to focus on a research question than a graduate student who's there all the time. And so I think about, well, is it actually a good training for the person who's going to be working on it? In addition to being an interesting scientific question, what are the skills that the person who is working on this will learn, and are those, you know, appropriate? And then also, is it something that we can get funded?
Of course.
Because the experiments cost money.
And the last question for us, but students and our guests can please ask questions because this is a very interesting conversation. I've learned a lot, and I share some insights that you're bringing us today. What is your advice to faculty members interested in developing a lab?
Oh, in developing a lab. Yeah, I think it's really, you know, what is the research question that you're excited about? I mean, you have to be excited about it in order to want to get up in the morning and work on it. So that's the number one thing. And then what can you do with the resources that you have available to you? And so, you know, what is the scope of the project? Where can you make a unique contribution? Yeah, I think, and then of course, finding people to work on it with you, getting equipment and things like that. I think, you know, all of our projects typically start pretty small, and then we see, okay, does this actually have legs? Is this something that's going to take off? So I think starting with a small question and getting something you're excited about and getting a couple of students excited about it is important.
Of course, and mentorship and networking, and all those things combined together.
Yes, there's a lot that goes into successfully running a lab, and those are all really important as well.
Great. Do we have any questions? We have one.
My best experience in the field of science. I mean, I guess I think back to certain experiments, like when I discovered that the demethylase DME was demethylating this imprinted gene. It was like the first time that demethylation had actually been discovered and the mechanism had been discovered in any organism. So that was very exciting. The other best times in science are like when a PhD student graduates from the lab and I get to introduce them and their talk, meet their parents, and you know, I feel very proud of the students who have come from my lab. So
great.
So the second question is, what are the qualities that you look for in someone for them to be productive in a sustained way?
The most important thing I look for, you know, when I'm thinking about bringing people into the lab is, do they have that innate curiosity and drive? If they have curiosity about how the natural world works, I feel like, and you know, it's just like they have questions they want to answer. That to me is the most important for someone being successful in the lab. Like it's really hard, like I can help with all of those other skills, like how do you ask a scientific question? You know, what's a good problem? How do you actually, you know, do an experiment? But if the motivation and curiosity piece is lacking, that's something that I haven't been able to, you know, to help develop if that isn't already there. So I think I really look for people that are very curious.
I have another question from an online viewer. It says, what was it about epigenetics that caught your attention to start working on? And what was it like working with your team during research process?
Yeah, I mean, I started working on epigenetics as a graduate student, actually. And it was one of those things where I was working on a particular mutant that had been identified in a genetic screen. And when we discovered you know, what that mutant was doing, it was involved in DNA demethylation. And so I didn't really set out, at least as a graduate student, to study epigenetics, but just through this, we were looking at some mutants that affected seed development. It turned out that was an epigenetic regulator. And so that's how I got into it. It was also at that time like a very, very hot, very new field when I was a graduate student. Which was between 2000 and 2005. And so there were some really exciting discoveries being made in plants and C. elegans, in many systems that it was, you know, it was just a fun field to be in. Like you would go to conferences and you would hear all this kind of crazy stuff, but people would be really people were really figuring out molecular mechanisms. Now I've talked so long, I forgot the second part of that question. Or something about my group.
The second part of the question. I forgot.
The second part of the question, what was it like working with your team during the research process?
Oh, well, that's great. Yeah, I mean, I love the people in my lab. And so, the way I run my lab is that, so there's about 9 people in the lab right now. They all have their own individual projects, but we all center around these themes of plant epigenetics. And so I really love lab meetings, group meetings in our lab where, people will talk about what they've been working on, and you can see these sort of ideas and synergies develop among people. I think a lab is a very fun place to be because it's a lot of talking about ideas and, how could we actually test this? And yeah, it's great. Don't let the haters get you down. I think, there is still sexism in science, for sure, but I think there's incredible opportunities. So I think, my advice for women in the field would be pretty similar to my advice for men. So, look for those research opportunities where you can. I had 2 children after I started my faculty position, which was definitely a lot to handle. And so I would also say, have a very supportive partner who understands the scientific lifestyle. So my spouse is also a scientist as well. And I think that was, he understood, when I needed to work on a paper when, he would help get me through those times.
Hello, how are you?
Hi.
Hey, what are the qualities of a successful student in your class?
Successful student. Well, as I said, so they're curious. They're also dedicated. So, they're willing to come to lab and put in the hours. I think resiliency is a big thing. So as I said, most of the time experiments don't work, at least not the first time, or hypotheses are wrong. And so I think being resilient and not letting an experiment not working prevent you from starting your next experiment. And then I would say another big part of success is being a good writer. So I think the most successful people from my lab can write really well, and that's something that you definitely can develop as a student. But oftentimes we think of science and writing as very different disciplines, but I think the best scientists are really good writers again, and really good speakers. So being able to communicate science is incredibly important. Gerardo? Undergraduates starting in STEM. I think, where science is right now, it's critically important. Let's say a biologist really needs to be very skilled also in bioinformatics. And so I think if you are starting in STEM, try and stay broad for a while and pick up those sort of if you can pick up skills in coding, for example, or scripting, that's really, really important because there's so much data out there now that in some cases you may have a question where you wouldn't have to do an experiment. You would just have to analyze data, genomes, or that's already available. And so I think developing those skills as a biologist are very important.
Well then,
Yeah.
Every day is not a good day, right? So when students are working with their projects and nothing is going well, how is your approach with the students, like maintain them working and that, like developing that resilience that you're
Yeah.
talking about?
I mean, that's definitely one of the, I don't think I have any magic answers to that. It's certainly challenging, as you say. I think sometimes small successes can go a long way. So typically with students, rather than just having one thing they're working on, I like them to have a couple of things, like a main project and a couple of side projects. So even if the main project is not working, you can still have some success in another area. And that, I think can help you. Like if you have something you can sort of, okay, I'm just going to set this aside for a little bit. I'm going to work on this, get some success. And then, you know, sometimes setting aside a project, you'll have more ideas. You'll have, you know, some ideas on how to overcome a roadblock. So I think that's important, working on a couple of things. Yeah, just finding small successes can help a lot.
well, we're very thankful and grateful that you're here with us and that you have this small but big information and motivation talk. So we would like to invite Dr. Aru for a gift as a token of our gratitude for you being here with us.
Thank you.
And we conclude, but please don't forget to complete the evaluation of the activity through the QR code or the link found in the program. We thank the National Science Foundation for sponsoring this activity.
Oh, thank you. Thank you very much.
Yeah.
Thank you all for coming. Yeah, come.
This is the toughest part.
Thank you.
As I was saying, we thank the National Science Foundation for sponsoring this activity. We also thank Dr. Juana Negron Berrios, Dr. Alok Arun, and definitely Dr. Mary Gehring and all our guests for participating in this activity. We also thank our collaborators from the Barranquitas campus for their support to this activity. We invite those present to stay. We have some refreshments outside. We hope you have enjoyed this conversation. We'll see you next time. Take care.